I'd like to begin the formal presentation with a short introduction of Applicon. Applicon is a world leader in developing and supplying advanced bioreactor systems from lab scale to production scale. And we supply cultivation systems for early stage initial screening throughout process development and up to full scale production. Applicon is known for bringing new technologies to the market, and these technologies offer advantages in process efficiency for research, for pilot plant and production. And the Ovisio I line that will be introduced today is an example of such a novel technology. The system has been developed by Ovisio and is marketed by Applicon starting today. I'd like to welcome our speaker for today's seminar, Professor Frank Guderman. He's a professor at the Fachhochschule Bielefeld, or in English, University of Applied Sciences in Bielefeld, Germany, and has been involved in development and commercialization of several cell counting systems. Frank, you may begin when you're ready.
So, okay, that's the introduction that is being cut off. So, hello everybody, and thank you very much for the invitation. And I was asked to tell you something about automation of cell counting and how differently we now view cell counting at all. So, and there I would like to start with a brief introduction of myself. Okay, this is only to prove that I did study indeed, and that I have did something with mammalian cells. We had the idea of changing the way of cell counting in 1995. 3 guys, 1 with the money and 2 with ideas. That was basically because cell counting was very bored. And it was dangerous to make mistakes because, as we see in my presentation, there is, a lot to it. We make a lot of decisions based on cell counting and viability, and therefore we developed something like the CDEX, and this is a cell counter, CDEX cell counter with a microscopic optic inside that takes images, stains the cells, so it takes away a lot of the manual handling and a lot of errors that can occur while handling. That was the first generation. Then the second generation looked a little bit nicer because we had the money to develop something, that was even from the handling better than the first version. And then while we found out that, and that may be the same with Ovisio, that the image recognition software is not so easy. It's sometimes tricky because what people want is one image recognition software for all the cells, for every cells, except for prokaryotes, for example. But when it comes to mammalian cells, it would be best to have one image recognition process for all cell lines, for all different cultivation processes, whether it's batch, repeated batch, fed batch, or perfusion. And that also makes a difference, because cells may look differently over time, and for the reason to fulfill those those customer requirements, we put very much effort in developing the image recognition process, but then we were limited to processes where the customer was able to take away 1 milliliter of sample for the recording of the cell concentration. And we said, well, there are thousands of customers using well plates or petri dishes or whatever. And then we developed something we called the cell screen cell imager, which was a system for well plates or tea flasks, whatever. The second generation cell imager was the Cella Vista. And then we had other product lines like the Cuda, a biochemistry analyzer that is able to measure now up to 20 different analytes in cell culture processes like glucose, lactate, a total protein, LDH, whatever. Then in 2008, we acquired a small German company, Schärfesystem, with a Casey counter. That's basically a particle counting system based on the Coulter counter principle. So with the deals with the electronic properties. Then in 2009, okay, this is the sales partner network. We had Roche acquired the company and made the investors very happy. But Roche found out very quickly that there was one product line, the biochemistry analyzer, which they had to replace, or they wanted to replace. And then a friend of mine and myself, we bought back this product line from Roche in 2011 and set up another company which is still working very well. We have two different analyzers now to sell the reagents, which is always a good idea, and do of course the GMP validation services and all the service for those instruments. But now, we want to come to manual cell counting and what it really means to count cells. In fact, the cell density or the cell concentration and also the viability, those parameters are linked to each other and are very important. It's not only cell concentration, but it's always viability that is important to monitor the status of the culture, to calculate different rates, production rates, consumption rates, but also the economics of the whole site, of a set of bioreactors where pharmaceutical companies produce pharmaceutical substances, and also, of course, to document cultivation processes for the FDA, EMEA, for the authorities. So it is very, very important that these data are reliable, of course. And in fact, it's the main reason why people have to be in the lab over the weekend, because you cannot take a sample, let it sit there on the bench for 2 days and calculate the cell concentration or get information on cell viability afterwards. So until today, the standard method is based on trypan blue dye exclusion, and we use this to monitor our processes. So, but how do we do this, and what are the pitfalls that can occur here? Usually we have something like a Neubauer chamber, a chamber with a defined area that is marked on the area, of one of those bars here. Then we have a certain depth of the chamber, but the depth may vary, and I come back to that later. And what is here part of the literature, you can clearly distinguish viable cells in light blue from dead cells in very dark blue, and then count those cells and calculate from the volume that the cell concentration and also the dead cell concentration and the viable cell concentration. So you can discriminate between viable and dead cells. But if you put a cell sample under a microscope, you will find that it's not so easy to distinguish viable and dead cells. And like this object here is clearly dead, but there are others like lighter blue ones here. And is this cell a dead cell or is it a viable cell? And one may judge this as dead because there's already trypan blue stain in it, so it has to be dying, in the process of dying. Or the other person may say, well, that's a healthy cell because this cell is able to keep sort of some percentage of the trypan blue still out, so it's still doing something. So counting cells and the errors that occur from cells are not human errors alone, like preparing the Neubauer chamber and having a particle of dust of 10 microns between the slide and the chamber. That would result in the worst case in 10% deviation of the cell count. But it's not that alone. It's simply that every user has a certain bias of how to count viable and how to count or to define dead cells. And that's basically why we feel and we see deviations between different operators if cells are being counted. And this is what we see here. We have here 4 different operators, and we gave them a set of samples, and they didn't know that 4 samples were the same, and asked them to count the cells and to write down and document the cell concentrations. And here, this is only cell concentration. It's not viability. And you see that the deviations are sometimes here operator 2 from an average of 800,000 cells per milliliter to over 1 million, more than 20% by the operator. And it is, even more surprising if you then repeat this experiment with the same set of samples with another chamber. So with another type of measurement, with another tool that you have. The only difference between the Neubauer chamber and the Fuchs-Rosenthal chamber is that Neubauer chamber has a depth of 100 microns and the Fuchs-Rosenthal of 200 microns. Why that is the case is really not so clear. But if you look at cells that are 15, 20, 25 microns in diameter, and if you have a chamber that is 100 micrometer of depth, the rim effects, if you have, because the cells, they are not actively going into the cell, they are driven by adhesive forces into this little tiny slit under the chamber. So there are rim effects where there's no movement, almost no movement of liquid. And this rim, if it is like 20 microns, and the bottom and the top of the chamber is 40 microns, so 40% in case of the Neubauer chamber, and it's only 20% in case of the Fuchs-Rosenthal chamber, because simply the height or the depth of the chamber is different, and that may cause deviations. But to be honest, I don't know if that's really the case. It's only, something to try to explain this. And we repeated this not only with our people at the university, because you can always say, well, are they well trained? We repeated this with lab technicians at different pharmaceutical companies. Okay, I cannot tell you which ones they were because they didn't wanted us to publish those results. Okay, so what do we get when we count cells? We get results that are subjective, user-dependent, with low precision and pure accuracy. That sounds almost perfect. Due to manual sample preparation, influence of the geometry of the tool we are using, individual image interpretation, and low statistical significance, because we are not able to count 10,000 cells under a microscope. This is also something where mathematical statistics plays a big role. So if we count 100 objects, mathematically we cannot be more precise than 10%. And that's simple mathematics. It has nothing to do with some sort of error we implement by using our hands to prepare the chamber and everything. Okay, so one may ask, is it now the year to change all this by looking into other technologies? No, of course not. There are many, many different methods and also instruments to measure cell concentration and also cell viability. We only see here a few of them, and I want to look into two different measurements or instruments, like the Cuda counter principle first, and then also flow cytometry, which seems to be a very, very good instrument, even to, or especially to distinguish viable from dead cells. And the Cuda counter basically measures the electrical resistance over a pore, and the resistance, if there is only liquid in this pore, is constant. If if a particle enters this pore, the liquid gets replaced by the particle. So the resistance over the pore is different, gets higher. And there, from this information, you can calculate cell diameter. To do so, the cells or the particles that enter the pore should be ideal round objects. If not, you get the wrong information. So that's one assumption you make that may lead to results that may not be that precise. And here from the literature, it's very easy. You get the viable cells because they have intact membranes. You measure the resistance change caused by the whole cell. In case of dead cells, the pores have holes. The membrane have pores and holes. So the surrounding liquid is able to enter the cell, and you only measure the very compact cell nucleus. Okay, so you get two different peaks for viable cells and for dead cells. But if you put cells into this instrument, the reality looks a little bit different. You get one peak, one very wide peak, so that shows you also the diameter range your cells are in, and then you get sort of a very small peak, tiny peak, sometimes only a shoulder that is attached to the main peak you get, and these are the raw data you get. So it's not an image like you look through a microscope. It is this diameter histogram you get, and you have to interpret this. And the interpretation leads you to certain size intervals where you say, for example, like in this case, 6 micrometers up to 14, 15 micrometers are the dead cells, and the viable cells are bigger than those. And this way you can discriminate viable cells from dead cells. But now the culture grows, and the conditions inside the culture vessel may be different from the start. And then cells may tend to get bigger or smaller, so you have to rethink your borders. So this way, due to the fact that you have to interpret the diameter histogram, you apply a human interpretation factor to this method. So it is not that automated as it seems, and it is really sometimes difficult to follow the cultivation process and get reliable data on viability with this instrument. Don't get me wrong, this instrument is perfect for particle counting, and it's perfect for cell counting. So if you focus on total cell concentration inside a bioreactor or inside a sample, that is really good because it counts like 5, 7, 10,000 objects per sample. So it's very reliable in cell counting, but viability is difficult. Okay, the second is the flow cytometer principle. And I think everybody knows that there are different dyes that can be linked to different parts of the cell. And those dyes are being detected. And here the difficulty of data interpretation is the same. It is a different approach, but what we see here that we have dead cells, necrotic cells, apoptotic cells, and they are clearly separated. So that this is a good tool to distinguish between necrotic and apoptotic cells. But the border between viable cells and apoptotic cells is somewhere, user independent, and you never know because there's no clear line, no minimum here in this cloud of individual dots between viable and dead cells. So also here it is difficult to distinguish and to get data on viability. So that means if you do not use the original standard method for whatever reason that has become the standard method with Trypan Blue, it is difficult to compare data from other methods, from other instruments to those standard methods because they rely on different physical parameters like resistance or on different dyes that are linked to different molecules inside the cell. And that makes it difficult. So automation of cell counting may lead to instruments that simply automate the manual way of cell counting with Nubar chamber and Trypan Blue. And there are some, and this is only a small small example of different cell counters. The CDEX Bicell of Beckman Coulter Auto T4 from Nexcelom that was based for the medical market and others. And what do these systems? They automate the process of staining. They transfer the stain sample into a flow cell, and a microscopic image is taken from those samples. So the analysis is based on the same information than the manual cell counting, the manual trypan blue cell counting, and the image is recorded with a CCD camera and then automatically analyzed by an image recognition software. So the whole process is automated and it is very transparent, and that's important. It is not like measuring something like conductivity or resistance or whatever. You have an image, that image is being displayed by these analyzers, and you have labeled cells which show the result of the image recognition process. So the user is able to gain confidence in this method and in this new automation. And what you see here, if you ask different operators to count cells, or if you have one operator who counts cells and ask this person to count more times, like duplicates or replicates of the sample, you'll get the usual deviations between operators or between different samples being analyzed. And what is very important, that you have a device that displays sort of an average of the data that have been acquired years ago. And so it is also important not to set a new standard, but to have something, an instrument that is able to be calibrated in a way that the data that are gained with this new instrument fit the data that companies presented the FDA years ago. Well, because it's not possible to set a new standard here, because you have to continue the story that a company told the FDA years ago. So what have we achieved? We have better precision and better accuracy in our samples or in our data that are being generated. But what we still have to do is we have to draw samples. And also there you can do many mistakes because it's a manual process. The sample needs to be stained. It's difficult to perform online measurements because sometimes, one measurement, one data point per day, as it is usually done with manual sampling, is not enough because you want to know more about your process. Sample volume is being consumed. Also, this is or may get an important factor while trying to automate offline counters. And that's what I named here online cell counting. It's the automation of the sampling procedure and to hook up a device that is able to count cells. Here, this is for a 40-liter stainless steel bioreactor. And this is an instrument or a device that takes sample from such a bioreactor from BTS, Biotechnology Services. They developed a valve here, which can be implemented into the bioreactor. It can be steam sterilized. That's important because there's no chemical disinfection or whatever acceptable from the customer. It needs to be steam sterilized because also that's the standard procedure. And this little tower here, that's the sampling control unit that is next to every bioreactor that is being sampled. What is it called here? The sampling process control unit. There's the computer that drives all the different processes that are done by the sampling device. Also, it's only for the sampling device. This is the distribution valve or the distribution unit. The sample sometimes needs to be transported over a distance of 20-25 meters from the bioreactor or from the sampling unit to this analytical unit because they may be some meters away, or the customer may want to connect more than one stainless steel bioreactor to this device because it's very expensive. And to somehow reduce costs, it may be worth to connect more than one bioreactor. But then, of course, you run into issues with cross-contamination because there's one valve that is here in the middle. Now, you can see it here in this image that connects different bioreactors, and that may not be so beneficial in pharmaceutical processes. But of course, that was the production type of thing from the same company. There's also a lab version, and you see right on the left-hand side, this is the bioreactor, and then you have the unit that draws the sample. Here you have the unit that filters out cells, because somehow once you have a sample automatically drawn, you may not only want cell counts and viability, you may use another device for biochemistry analysis of glucose and lactate, for example. And you end up with a setup like this. And there are also other companies that provide those setups, but they all have certain downsides because the consumption of sample volume is now very, very interesting. Because why would you spend the money on an automated sampling device to draw sample more than once a day, like every hour, every 2 hours? And it's not only the sample volume that is being used for the analysis, it's also the sample volume that is necessary to flush the lines. You want the sample to be representative. And the same then what is in the bioreactor. Okay, so you need like 15 to 25 milliliters of sample volume per sampling. And that means if you sample like 10 times a day, you have 150 to 250 milliliters of sample volume to spend. So in a 2-liter bioreactor, that would take you 4 days and the bioreactor is empty. And that's the point, consumes a lot of sample volume. It is expensive because you hook up by a sampling device also one or more analyzers dedicated to this one process. So it's not only the sampling device that is expensive, but also the analytics that you want to put behind, like an HPLC system, cell counter, whatever. Maintenance demand, of course, if the analyzers run all day, then you need a lot of maintenance. The cross-contamination that I mentioned before, if you connect more than one bioreactor, and you need a lot of bench space. Next to every bioreactor, really, really a big system. And there is another thing, which I only want to mention briefly, what is called inline cell counting. So there are other devices that can be mounted directly into the bioreactor. Here, like turbidity probes, for example, which measure turbidity, but don't give you any idea of viability, for example. Then laser reflection analysis. This is something that now these days enters the market, where you get the reflection signals of cells from a laser that is rotated inside the bioreactor, and you analyze those and get information on particles from 100 nanometers to centimeters. And that's exactly what makes it so difficult to adapt it to cells, because the cells are in a certain range of dead cells, 5 micrometers to, let's say, 50 micrometers viable cells, and you only want to measure those particles, and then you have to rethink the data you get and interpret them in terms of dead cells and viable cells. The electric spectroscopy, for example, measures viable cells, and in theory doesn't give you any signals on dead cells. And there is something, the in situ microscope, which has the basic principle that you mount a microscope into a bioreactor and then count cells and distinguish viable and dead cells by something called dark image spectroscopy or dark field spectroscopy. But this is a prototype. It is still at the University of Mannheim and never really made it into the market. So to be honest, I don't know why, because it looks logical, but there seem to be other issues with this technology. Okay, advantages: no sampling necessary, so you don't waste any volume, and the data are generated continuously. So even better than online sampling every 2 hours or every 4 hours. Downsides: viability is difficult. The results are very, very difficult to correlate to the standard methods. Cells may adhere, so there's sort of the surface fouling, which may change the signals, and also it's again expensive because you need one device for every bioreactor. So one could ask, what's next? So what is the out, or what is a new technology that may be worth looking into? And that is a device, the Inline F, of a company named Ovisio, and the technology that is built in here is holographic imaging. The main achievement here is that there's a label-free analysis possible, so it is non-invasive. You don't have to stain the sample, and you can put it back into the bioreactor. So you don't have to waste any money, any cell volume, sample volume. And the fact that there is no staining procedure necessary, that is the main benefit, because you have sort of a closed loop between the process, the bioreactor, and the analytical part of the system. You have no sample preparation, no toxic waste with Trypan Blue, real-time imaging, and also refocusing. This is not the first imaging system, of course, like the CDX or the Weissel of Beckman Coulter, the first ones on the market. They also use image data to do the analysis. But when something goes wrong with the autofocus procedure in these systems, you are really in trouble, because you cannot trust the signals anymore, you cannot trust the data anymore, and you have no way to get hold of any information about the process, because the images are not focused, so they cannot be analyzed, and the information is gone. In case of the holographic imaging, that's different, because even if the images are slightly out of focus, you can refocus on the holographic information that you store on your disk. So it is not only an image that you have, but the holographic information contains more information than an image, but only occupies the same disk space. And also that may be very important. And you get quantitative results, so no raw data here to the system. The connection between the bioreactor and the measurement, the analytical unit where the hologram is generated, that's a disposable part. And the customer should expect that replacing a disposable part and starting a new process should give the same results if we have one correlation factor, and it should not change the correlation factor. Here you can see this, the correlation between CDEX and manual data using a factor that's usually 0.83, 0.85 or something like that. For whatever reason, it is there, but if you multiply the results with this factor, you get the same also over a long period of time. And now the correlation with the new system. Here we see the many dots compared to the few manual dots here in a process that run 14 days and run through all different stages. We have the viability in the beginning of the culture. This is the viability reading, 90%, and then the viability drops down to 30-35%. And the different methods like CDEX, Newport chamber, even Casey, are almost in line. Casey deviates a little bit here and shows a little higher viability. I go back again here, that the correlation between the Inline F, the new technology here, and the CDEX is not 100%, but implementing a correlation factor here, you can see that's a different cultivation here. With a growth phase for the first day, and then a dilution step here, and then the culture grows again. So with the correlation factor here, we are able to set the manual cell count and the data generated with the holographic images perfectly together. And that also is true for different batches in different bioreactors at different dates, and with different, you see here in red, BioConnect A, BioConnect B, with different BioConnects. It looks like the BioConnect B is not as good in the end of the culture here, days, let's say 8, 9, and 10. But this is due to the fact that we run the BioConnect, which is a disposable out of specs here. So the vendor of the BioConnect clearly told us that the BioConnect is good for 3 up to 4 weeks. And this was here the week 11 when we used the BioConnect. So clearly out of specs. Why we have the deviation here in cell concentration is not really clear, but if you drive something out of the specification, there should be a reason why there's a certain limit to use this. So in summary, the Inline F system generates the numbers, and that's very important automatically. It's an online measurement that is not consuming any sample. The sterility of the culture process was not affected, although we used the BioConnect longer than suggested from the vendor. It can be adjusted to the CDEX, to the manual cell counts, whatever you want, by one factor that has to be implemented. You can refocus the images, which would be very nice, I think, for FDA purposes. If there are some questions about harvest times or whatever, or feeding points, you don't need to use any Stripe and Blue, and it's easy to operate the system, because you only have to mount the system, the BioConnect, to your bioreactor. Everything is autoclaved, and afterwards you can run the bioreactor as you're used to. Only put the chamber, the counting chamber of the BioConnect into the holographic analytic device, and then you can run your process.
Okay, that was the presentation of Frank Gudeman. We would like to thank Frank Gudeman for his input. For now, we will actually go to a short presentation from Philippe Mathuis from Ovisio, and he will show us some more details on the system itself from Ovisio and the options that are available with this system. So whenever you're ready, Philippe.
Okay. Good evening, everybody. So my name is Philippe Mathuis. I'm founder and co-founder and CEO of Ovisio Imaging Systems, and we have developed a platform technology that we specifically use in bioprocessing for applications with suspension cells and adherent cells. And the system we have shown here in the presentation is our bioreactor monitoring system. So that system consists of apparently there seems to be a small problem with the presentation. I'm sorry for that. I will relaunch it. So a short intermezzo. Okay. So the system consists of a disposable probe. That is, we call that the BioConnect, and this disposable probe is consisting of a reading cartridge, which is a flow cell, and it comes combined with a reusable pump. And the idea is that the system is assembled, the probe gets inserted into a standard slot of a bioreactor, and then the pump is connected. The reading cartridge is connected to the Inline F microscope, so that is the microscope for this application. And from that point in time, cells, the pump is starting, and cells are pumped out of the bioreactor. They run through the reading cartridge, where the microscope will compute the cell count and cell viability based on a holographic fingerprint. And this is a closed loop system, so the cells flow back into the microscope. So, and the system, in fact, computes cell viability based on a holographic fingerprint, and we use several parameters to compute this viability, but we have benchmarked it with the current gold standards, and it gives the same results. So, and this measurement is based on the lens behavior of a cell. Let's say, to simplify things, that a healthy cell will, or a live cell will create a light cone, so will concentrate the light in an out of focus point, and a dead cell with a porous membrane will diffuse light, and we combine that with the dry mass measurement of the cell and about the average size, etc. So we use a certain number of components, and you also have we also have a software that has similar functionality as a flow cytometer. So the green dots here, they represent the living cells. The red dots represent the dead cells, and you will also see yellow dots, and they are on cell aggregates. So we also measure the number of cells that are aggregated. And the difference between a classical flow cytometer and the blood charts created with our system is that when you click on a dot, you'll have the corresponding image of the cell. The system also draws automatically the growth curves of the bioreactor. So you see we have much more measurements. We have a measurement point almost or every 30 minutes. So every 30 minutes we take a sequence of 25 holograms, and that allows us to compute in a very accurate way the growth curve of the bioreactor. We also have access to data such as the average cell diameter, the cell size, the cell optical height, and the cell volume. This is the microscope. So that is a specifically adapted microscope for the online cell monitoring application. So it does cell count, viability, morphology, it measures aggregates, and in the future will be able to be used to predict optimal harvest times for the cell. So the analysis time is very fast, and we acquire holograms, as I said, every 30 minutes, but that is a software parameter, so it can be configured. And the current range is from 1,000 to 12 million cells per milliliter. And we are working on increasing this cell density to more than 100 million cells per milliliter. So the cell size that we typically measure with this microscope is from 2 to 100 micrometer, but that can be adapted in function of the needs. So the system also comes with this disposable, which we call the BioConnect. So the disposable is tubings with a new pump that we have developed. It's a membrane pump that is like a heart, so with an inlet and an outlet. The throughput is 4.8 milliliter per minute, and the cells, they stay on maximum 2 minutes within the fluidics. The pump is strong enough to evacuate possible clogs or possible obstacles or air bubbles that could form within the flow system. And it's made from an autoclavable material. The tube length for insertion into the bioreactor can be chosen and customized depending on the bioreactor type that is used. And the typical bypass volume, or the volume that is within the fluidic system, is between 3 and 8 milliliter, depending on the fact of if the pump is pumping or is not pumping. So if the pump is full, it's 8 milliliter. If the membrane is empty, it's 3 milliliter. As I said in the beginning, we also have applications for adherent cells. And here you see an example of a system that we use to look to through multiple layers of, for example, cell factories and hyperflasks. So it's the same technology, but then used for adherent cells. And we also have an application where we look through a 200-layer bioreactor that is also made for adherent cells. So basically, Ovisual has developed a platform technology and that we mainly focus on applications within the field of bioprocessing. So we have solutions for suspension cells and adherent cells, and the technology is fully label-free. So it does all these computations fermentations without staining, without dyes, and it allows us to automate manual work and to have feedback loops between the control systems of the bioreactor. It's totally non-invasive, and it's very convenient and easy to operate and to use. So if you would like to have more info on this specific product, please go to www.eline-f.com, or if you want to try the system, please send a mail to info@orvisio.com. Martijn, back to you.
Okay, thank you very much, Philippe. Now we will move into the Q&A session, and at this point I would like to invite our audience to continue to send questions or comments using the questions panel on the right side of your screen. We have received a few questions, and if you have any questions about the system itself from Orvisio or on the previous presentation of Frank Gudeman, please enter those questions. And I have one question here that I think is for Frank Gudeman. The question is, how do you validate your method with the CDEX high-res, for instance?
So I think the question is, how do I validate a new method like the Inline F with the CDEX or with the CDEX high-res, and this is that we take the same samples out of a process, and it is important not only to have like one or two samples, but to have samples from different states of the culture, so in the growth phase, linear phase, stationary phase, and even in the dying phase of the bioreactor, and then use the data of the CDEX and correlate the data generated with a new method. And if you do so for many processes over time, like if you collect data from different bioreactors this way, you get a good impression of what the correlation factor for the new method, whether it's the Inline F or any other method is, what the correlation factor looks like.
Okay, thank you very much. A second question that pops up. What kind of cells has it been used already, and is it also feasible for microbial cultures?
So I guess this question is for me. So for the time being, we have mainly focused on mammalian cells, and we've tried it on the majority of cell lines used within the bioprocessing industry, such as CHO cells. So we have done the vast majority, but the system can be easily adapted to and trained to recognize a new cell line. We have not launched the system for bacteria yet, but we have a research project that is really focusing on bacteria and recognizing different sub-species of bacteria. And that is something we intend to launch in the relatively near future. Perhaps a last comment. We have also a project within a brewery here in Belgium where we use the technology to measure fermentation of yeast cells in beer. And the algorithms that we have used for bioprocessing, they are also valid and usable for yeast cells.
Okay, another question. Is there any hydrodynamic damage on the pumping of the cells through the tubing?
So this development took quite some time. It took more time than we initially planned. And one of the reasons is that we had to design a new pumping mechanism. So we decided not to use peristaltic pumps, because we've used them in different test setups we made when we were launching the company, and we saw that the peristaltic pumps, they had some impact on the cells, and the cells don't really like peristaltic pumps very much, and that's why we came up with a new design, which is a membrane-based pump that has an inlet and an outlet, and so functions in the way of a heart. And we have done many tests and determined that this pump system has no impact at all on the cell morphology, and cells remain happy when they are in the tubing and run through the fluidic system.
Okay, and then for the last question of today, how can the equipment differ between viable and dead cells?
So we have patented a method, and the basic principle, so with holography we have the capability to refocus objects that are within the hologram, and a hologram is typically a cube with sides of about half a meter. So in fact what we do when we measure viability, first of all, you will see that healthy cells, when they have an intact membrane, they will behave like a lens, and they will create an out of focus light spot, so a very tiny spot, where a dead cell that has a porous membrane and is losing its cytoplasm, that will diffuse light. So we combine this, let's say, fast method with the total volume measurement of the cell. We use the average diameter of the cell. We measure the position of the nucleus in comparison to the cytoplasm. So in total we use about 16 different parameters to define this viability. But we have of course tested and validated this viability measurement with the current gold standards, and it's the same as the current trypan blue exclusion-based methods. And I would even say more, is that for some of our clients, we have seen that cell death is detected about a day earlier than traditional trypan blue exclusion-based methods too. And we think that is because we see changes occurring in the cell before the cell dies. And moreover, this is one of the features we are going to launch in the future. That is the possibility to define a holographic fingerprint specific to apoptosis. And holographic fingerprints, this concept, in fact, it's like a Shazam of a cell. So we compute this fingerprint, and based on that fingerprint, we can say which type of cell it is. We have an application where within white blood cells, we can detect the different sets of cells and also determine their viability. That's it.
Okay, then, well, we have one more interesting question that came in. This is for Frank Gudeman. What are the advantages and disadvantages of biomass monitor based on connectivity and capacitance?
That's a good one. Connectivity and capacitance. The advantage is that this is an inline method, so you can mount a probe directly into the bioreactor, sterilize it in line with the process or with the bioreactor in the autoclave. So it's very easy, but you are using capacitance of a cell. In theory, you're measuring all the viable cells, but you are not measuring cell numbers or concentrations directly. What you get is a reading on cell volume, total cell volume, that is in a certain space close to the probe. Everything is fine if cells don't change the diameter or its volume. So if you would have the same number of cells, the same cell concentration in a bioreactor, but the cells, for whatever reason, double their volume, this probe would give you an increased signal, which would lead you to the conclusion that the number of cells in your bioreactor, in your process, is increasing. So you have to take care about changing diameter over the culture period or over the cultivation in your process, and you have to think about this. Nevertheless, if the cells are very healthy all the time, are kept in a certain status of the culture, the method works well. But if you do like fat patch cultures with decreasing viabilities, the interpretation of the data is a little bit tricky.
Okay, thank you very much. That is all the time we have for today. If your question was not answered, we will email you a direct reply. I would like to thank everyone today for attending today's webinar. Don't worry if you have missed some part of the presentation. Simply visit www.applicon-biotechnology.com, where our webinars will be archived. If you want to share this presentation with your colleagues, simply email us their name and email address, and we will send them a link to the archived presentation. Please join me in thanking our speakers of today, and thank you all for your time. We hope you found this webinar informative, and have a great day.